With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello students, so today we have a question here is ligase enzymes are used in genetic engineering to cut open plasmid DNA, insert plasmid DNA into the bacteria, isolate the DNA making up a human gene, join human DNA to plasmid DNA. So these are the options that we have with us and we have to choose the correct option. So what are these ligase enzymes and what is this genetic engineering? So this genetic engineering is the process of using DNA recombinant technology, DNA recombinant technology. So why this technology is used? This technology is used to alter the genetic makeup of the individual. So this is known as genetic engineering. So let's talk about our options. Why ligase enzymes are used? So these ligase enzymes are the enzymes that are used in this DNA recombinant technology to join the fragments of the DNA of human and plasmid DNA. So this technique involves enzymes involves the joining of these fragments of DNA, and then this recombinant DNA is produced. So it is also involved in the ligation of vector insertion. So this is also involved in the insertion of plasmid's DNA. So the next we have is this ligase enzyme. These works on the blunt ends of the DNA. So what are these blunt ends? These blunt ends are the ends that is 3' hydroxyl group and 5' phosphate group. So they will act on only this type of strand, that is it should have 3' hydroxyl group and 5' phosphate group. So this is the function of these ligase enzymes, that these ligase enzymes involve the joining of the fragments of the DNA, and it involves the joining of DNA into the plasmid's DNA. So the next we have is it also involves the insertion of plasmid DNA, and it works only on the blunt ends of the DNA, that is 3' OH and 5' phosphate group. So the correct option we have is the last option to our question, because last option we have is join human DNA to the plasmid DNA. So this is the correct option, and this technique involves the recombinant technology, that is to make the recombinant DNAs by joining the fragments of different DNAs from different sources. So the first option is cut open plasmid DNA. This is the incorrect one, because it do not cut any enzyme, it joins the fractions or the fragments of the DNA. So the next option we have is inserts plasmids into the bacteria. This is also the incorrect one, because this technique do not involve this kind of things. The next we have is isolate the DNA making up of an human gene. So this is also not involved, because isolation of DNA is done by specific DNA receptors. So therefore this option is also the incorrect one. So the correct option is last option to our question.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.